we have a $7 million NSF grant in which we're connecting with middle school teachers. They come into our laboratories and do science with scientists. We have 4 projects, each is led by a postdoc. And basically what we have are middle school teachers who are co-creating and working on real scientific projects with scientists. And then they're turning that into lesson plans where they and their students in the classroom can make original discoveries and be real scientists as well. And so, let me hear it. So we have our 12 teachers who are here. Let me hear our teachers. And Monday we heard from Team Dirt working in the genomics lab. Tuesday we had heard from Team Jaws working in the paleontology lab. And if you can't tell by their attire, we're going to hear from Team Ant working in the biodiversity lab about their project. So if you please come on up, Juliana, Kelly, and Cooper, and share with us what you've been doing.
All right, everybody, before we get started, we got to see your ant sign. Everybody, let's see, hands up balcony. Come on, ant sign, there it is. All right, welcome to Team Ant. First member.
Oh, hi, I'm Juliana Martinez, and I teach 7th grade science at Westmore Middle School. My name is Kelly Wall, and I teach 8th grade science at West Millbrook Middle in North Raleigh.
I'm Cooper. I teach 7th grade social studies, language arts, and science in Pender County, North Carolina.
During a very long, hot summer day, a young queen ant flies into the bright blue sky. She's on her way to begin her mating flight. She flies higher and higher into a cloud of all male ants. She will mate in the air. Afterward, all of the male ants will die, and the young queen returns back to the earth. The queen ant no longer needs her wings, so she breaks them off by rubbing them onto the ground. Then she starts digging a hole into the soft, damp soil. She's building a nest because she's ready to begin laying eggs. This will be her only job for the rest of her life. She will never leave the nest again. The queen is extremely vulnerable at this stage because she is all alone. There are many dangerous obstacles up ahead for her. Some of the challenges that she will face are predators. She'll also have to compete for resources, and she will be exposed to pathogens. Our team, Team Ant, is the most interested in understanding the relationship between ants and pathogens, particularly fungi. So why are we looking at ants? Because ants are abundant, we can find them anywhere, in cities, in forest areas. Their physiology and the way that they behave is all sensitive to the environment. The activity that ants perform, I guess, in the ecosystem is, I'm so nervous. It's so important because it has effects on other processes in the ecosystems, such as when they're building their colonies, they're overturning the soil, refreshing the nutrients, especially for a plant group. Shout out to Team Dirt. They are also scavengers. In this video, this was taken by our postdoc. You can see that they are struggling to figure out how to move this insect because they're trying to take it back to its nest. They eventually are able to move the entire thing with just about 4 ants. Like humans, ants are living in high densities with other individuals. They also have challenges of obtaining resources in order to support such a large population. And because they live in such small quarters in their colonies, they are very prone to disease transmission. All right, so this brings us to our big question. What is the relationship between human-driven environmental change and the ant pathogen community? So the very first thing we did was we went out to Prairie Ridge on our very first day and we collected this sample of monomoria. And that is small black ants, and what is so interesting about that colony is that they have multiple queens. And we can tell which ones are the queens because they have wings. And then on the right-hand side, we have our School of Ants project where we laid out pecan sandies around the museum to check the species diversity of the urban ants around the museum. So once we had our ants, we brought them back to the lab and we did 3 immunity tests on the ants to start testing their immunity to their fungal communities that they're living in. The first one we did was a behavior study, and we placed the ants in petri dishes for 10 minutes to see what kind of behaviors they're having. These behaviors can help reflect their immune system functions. So we're looking for 4 main behaviors. The first one is self-grooming, where the ants groom themselves, grooming each other, antenization, which is where they're touching antennas, and we're also looking for trophallaxis, which is where the ants are sharing their social food. Next, we froze the ants and we took pictures of their thorax and head to get a cuticle melanin reading. We're trying to see if there's some kind of trade-off between the ants investing melanin into their cuticle or saving the melanin. And the last thing we did was grinding the thorax and gastrus in a PBS solution, which will later be plated. And once we get the plate analysis from that, we will have a big broad picture of the immune function of the ants. But our big question is about the ants relating to their fungal pathogen community. So what we had to do was collect soil samples from around where we collected our ants. We homogenized the soil samples and we dated them with 3 mealworms. We were very careful to keep everything sterile so that we knew that the fungal pathogens that we were isolating came from the soil samples. So once we had those mealworms, if they died, we put them into a growth chamber after we sterilized their exterior to see if they would grow any pathogens. From there, we will be taking the pathogens that grow and putting it in agar. This is an example that one of our students from NC State, Michelle, this is an example of the pathogens that she was able to isolate.
Incredible journey, ladies, so far, hasn't it? What does this mean for our students, though? How can we take this incredible opportunity, this incredible experience, and get that same kind of magic in our classroom? When we were putting together our mission statement for our website, Kelly said she wanted to ignite her students' curiosity. Ignite her students' curiosity. I think that's the heart of what we're after with this program. I think that is the most essential part that we can do. To be honest with you, I have students in my classroom that aren't curious. We need to fix that. So as we started putting our lesson plans together, a couple things drove us in making sure that they were accessible to everyone. We used language that is readable and accessible to as many teachers and students as we could get. We also figured out how to take some of our high-tech lab equipment and make it cardboard science, where we can recreate this in the classroom. On the left-hand side, you'll see our highly technical ant behavior study chamber, cardboard box, and wrapping paper. In the middle here, you see our light box that we created for taking those pictures of the ants. And we also tested this light box. You see we got our lab camera here. We also tested the light box with iPads and iPhones, and the quality was good enough for us to use those observations as well. So again, repeatable in the classroom. And here is our official ant collection and habitat device. All of it came from the dollar store. But the most important thing, I think, we want to take into this classroom, again, that curiosity. We want our students to start thinking like scientists and behaving like scientists. I'll tell you a quick story. I live in Wilmington, and last week my wife joined me here. And on our drive home, we stopped about halfway, about at Warsaw. Went to Wendy's, parked in the parking lot. Wife ran inside, and I ran to the curb. Well, I guess she was enjoying her burger because it took about 15 minutes to come out. She finds me crawling around in the parking lot. Now, the good people of Warsaw at this point had started gathering a little out of the picture. And she came running out. She said, "It's okay. He's a scientist. I'm a scientist. What I mean by that is I see science everywhere now. I'm curious about the world. That's what I want to bring into the classroom for these kids. Now, one of the ways that we're going to do that is by having these kids actually be able to contribute to a citizen science project where they will be collecting data, observing behavior, and contribute to our mentors research. To talk a little more about that, I'd like to introduce Dr. Deanna Beasley.
Thank you for the introduction. I'm Dr. Kenan Fellows, and just want to say it's been amazing working with these teachers and just seeing the inspiration in how they interact with students and how they interact with each other. We're definitely motivated to bring science to the community. And I'm also motivated not only by studying, the impacts of these huge environmental changes on insect populations, but I'm also motivated to engage as many people as possible in our research. And in fact, so far, this research has involved a number of students already, students from North Carolina and Shaw University, and they've been a big help, not only in working with the teachers and helping with this ambitious project, but it also keeps them motivated, especially when the science gets a little tough. But it's, I feel like it's important to engage as many people as possible in research, and so I'm happy that we'll be working together throughout the year to bring that to the classroom. This is an ambitious project. We're interested in looking at insect community and behavior and pathogens across a large landscape, so we'll need as many classrooms engaged as possible to achieve that. And if we do come up with a data set, this will be so cool. So I'm glad to be working with such an ambitious and hardworking team, and we will complete. Follow us on Twitter, and have a website.